Thank you, Dr. Wen, for a wonderful presentation. Now, I'm going to talk about the challenges and promises of in vitro release test, that is IVRT, and in vitro permeation test, that is IVPT, to support bioequivalence of topical products. Before going into the details of my presentation, I have the following disclaimer. This presentation reflects my views and should not be construed to represent FDA's views or policies. I will start with brief background on the studies followed by common observation, that is from the submissions of ANDAs to the agency, then specifically the observation related to IVRT and IVPT, and end with concluding remark. So let me start with background. I would classify the methods to establish bioequivalence for topical products in two major categories. Traditional methods, which includes PK, PD, and clinical endpoint studies, and alternative methods, for example, modeling and simulation, and in vitro characterization based approaches. From this, I would like to further discuss about the observations noted from past submission on IVRT and IVPT studies. Let's begin with the promises of these methods. These methods are well defined, robust, and reproducible once properly developed and validated. Allowed to assess different formulation by evaluating the impact of process and quality control changes along with any post-approval changes to excipients. They are relatively cost-effective and time-saving. They can be automated. Useful to demonstrate batch-to-batch uniformity or variability. Now let's have a closer look at the comparative parameters of IVRT and IVPT. For IVRT, the study is conducted using synthetic membrane compared to IVPT, in which human skin is used. For IVRT, the dose is mostly infinite and occluded, whereas the dose is finite and unoccluded for IVPT. Mostly, hydroalcoholic mixtures are used as receptor solution in IVRT, where the released drug amount is in microgram to milligram range, compared to IVPT, where the physiological media with antimicrobial agents are commonly used as receptor solution and the permeated amount is in picogram to nanogram range. The release rate is the endpoint for IVRT compared to flux and cumulative amount for IVPT. IVRT data are relatively consistent, however, intra and interdonor variabilities are common in IVPT. IVPT data, unlike IVRT, may be used for IVIVC, which may potentially mimic the in vivo performance. Let's focus on the observations which are common in IVRT and IVPT from the submissions. I would like to share what is noticed in terms of study reports. Most of the time, the reports are combined across different sections. For example, analytical method validation combined with either IVRT or IVPT method validation. Generally, each study report should be submitted separately for each section. For example, analytical method validation, IVRT or IVPT method development, validation, and pivotal study reports. Now, let's take a closer look at the content of the reports. In general, the study reports should contain detailed information for experimental setup, which include but should not be limited to the treatment or handling of the membrane or skin prior to dosing, how the membrane or skin is equilibrated, about the randomization of dosing and blinding procedure, step-by-step dosing procedure, and how the dose is synchronized with sampling times, details about occlusion and how it is achieved versus unoccluded dose, sampling techniques, that is automated versus manual, environmental control data, that is the temperature and humidity during the conduct of studies, dates of experiments, handling of samples, for example, the condition, that is temperature and duration of sample storage, sample analysis, for example, detailed dilution procedure as applicable, data analysis, the detailed information on the calculations for release rate for IVRT, while flux and cumulative amount for IVPT, as well as the raw data, chromatograms, related SOPs, protocols as applicable. What we have discussed so far is about the common observation, and now I'm going to focus on IVRT and IVPT studies individually. Let's begin with IVRT. The question may arise, why the detailed information on IVRT method development? The main purpose is to know all the possible attempts made to select and optimize the specific IVRT method parameters for the specific drug product with adequate discriminatory capabilities. The developed method parameters should be used to design the study and protocols for validation and pivotal studies. What is noticed from the submission is that there is insufficient information or data or explanation on the selection of receptor medium and solubility of drug, diffusion membrane and its inner nets and binding, amount of drug product in the donor chamber, sampling times, study duration, stirring and agitation rate. I will further discussing this parameter in detail in validation section. Once the method is developed, it is important to validate or qualify the method in terms of apparatus, methodologies, and study conditions for the specific drug product to compare the test and reference products in pivotal studies. What is observed from the submission is that there are missing data or information to support IVRT apparatus qualification. For example, the empirical measurement for diffusional area of orifice and the volume of receptor solution compartment for each diffusion cell carried out at the in vitro base site, as well as the data available from the manufacturer if provided. In terms of study duration, USP Chapter 1724 recommends IVRT study to be conducted for at least 4 to 6 hours. However, it has been observed that the study is conducted for short study duration, for example, less than 4 hours, without any supportive data or explanation, which cannot be considered adequate compared to recommended in USP Chapter 1724 about 4 to 6 hours. Additionally, there is inadequate information or data for intra and inter-run precision and reproducibility. If an IVRT method is robust, it should be able to identify minor variation in the experimental parameters or setup, for example, temperature, dose volume, receptor solution, and mixing rate, do not significantly impact the release rate of drug. In that context, it is noted that the data are either inadequate or lacking this information. The method discrimination parameters such as sensitivity, specificity, and selectivity are crucial to support the validation of developed IVRT method. The most common observation related to these parameters include the selection of strength. For example, the highest strength should be 150% label claim and not higher than that, that is 200 or 250% label claim. To support selectivity, the statistical analysis should be performed using Mann-Whitney's test per USP Chapter 1724 using appropriate study design, for example 6 by 6 cells. Please note that the altered strength should demonstrate inequivalence compared to nominal strength using Mann-Whitney's test with adequate study design and number of cells. It has been also noted that the submission lacks data for supplemental selectivity, which is carried out using the same strength of the drug product as the reference product. However, the formulation will be designed in such a way that it provides different release rate. This parameter validates that it can detect differences in the release rate associated with characteristics of the formulation but not the strength. There are instances where the information about the composition and procedure for preparation of the altered formulations are missing in the submission. With that, abiding the timeline for my presentation, let's move to IVPT now. I will start with method development. As I briefly discussed the purpose of method development in IVRT, I will not go in detail about it since the same principle applies here as well. In that context, what is noticed from the submission is that there is insufficient information or data or explanation on the selection of apparatus, dose amount, study duration, sampling times, receptor solution and solubility of drug, skin source, skin type, skin preparation, and its barrier integrity test along with acceptance criteria. I'll be further discussing this parameter in detail in validation section. Quite similar to IVRT, same principles apply to IVPT validation as well, and I will not go in detail in the interest of time. Similar with IVPT apparatus qualification. Let's move now to the next validation parameter. From the submissions, the unconventional dosing procedure is observed. For example, apply the dose on the skin while it is not mounted on the vertical diffusion cell. Also, there is insufficient information or data in terms of detailed dosing procedure. For example, accuracy of dose applied, dose spreading procedure, and other related aspects to better understand the actual procedure. Another parameter is receptor solution qualification. For hydrophobic drugs, the chemical agents may be added to enhance the solubility in receptor solution. However, in general, it is not recommended to use the chemical agents which may have potential to alter the permeability of the skin. In addition, to prevent potential bacterial decomposition of the dermis or epidermis in the diffusion cell, an appropriate amount of antimicrobial agent should be used. It was noted that the amount of antimicrobial agents used in the studies are 5 to 10-fold lower than the recommended amount without providing any supportive data. Moving on to another parameter, that is skin qualification. For IVPT study in which the membrane is the human skin, needs to be appropriately qualified due to inherent variability associated with it. The submission sometimes does not have the detailed information about the skin handling during procurement and shipping, skin processing, for example, dermatome or heat separated epidermis, skin storage conditions, number of freeze and thaw cycles prior to use for tasting. Another observation is about the use of different anatomical sites for validation versus pivotal studies. For example, validation or pilot study carried out using back region skin, whereas for the pivotal study, the skin from the thigh region is used, or vice versa. To add more about the other aspect in terms of skin qualification, I would like to make a note that there are instances where the data are not sufficient to support the proposed acceptance criteria for skin barrier integrity testing along with the detailed procedure. In general, the acceptance criteria should be derived in such a way that it should differentiate intact versus impaired skin. Skin equilibrium with receptor solution is another important step that has to be performed before dosing. It is recommended to measure the skin temperature of the non-dose soil periodically to confirm the control of skin surface temperature throughout the IVPT study. Detailed information should be provided to support sampling and replacement procedure at each sampling time point. Each and every validation parameter is important. However, the method discrimination parameters such as sensitivity and selectivity are more crucial to support the validation of the developed IVPT method. In this regard, I would like to start with IVPT sensitivity. It is noted that there is inadequate information in terms of number of donors. For example, use of 1 to 2 donors which are not sufficient enough. There are recommendations about different approaches to demonstrate variation in drug permeation profile. For example, altering the dose amount, that is, if the nominal dose is 15 mg per centimeter square, then one lower dose and one higher dose should be selected. Another approach is altering the dose duration, for which one shorter and one longer dose duration should be selected compared to nominal dose duration, and similarly using the alter strength. I would like to make a note that the permeation profile using optimized IVPT method parameters should be compared using specific approach as discussed earlier to demonstrate higher and lower amount of drug permeated from the same donors. Please note that the specific approach should be identified during the method development phase for drug product under study, as one size fits all does not apply here. Now let me move forward with IVPT selectivity or pilot study. In general, the IVPT selectivity or pilot study should be carried out as a parallel assessment of taste, reference, and alter formulation, for which the taste and reference product should have similar permeation profile, whereas the reference and alter formulations should have different permeation profiles to ensure that the method is selective and discriminatory. Similar to IVPT sensitivity, less number of donors, for example 1 to 2 donors, are used, which is not sufficient. The data generated from this study should be utilized to support IVPT permeation profile and range and IVPT precision and reproducibility, for which it is noted that the submitted data are not in appropriate format. Also, the composition and procedure for preparation of the alter formulations are missing. Once we have the data of the study, the next step is about the analysis of the data. In this regard, it is noted that inappropriate approach to calculate flux is used. For example, the nominal time points for each sampling time point are used rather than the duration between two sampling time points. In many instances, there is removal of certain data after sample analysis without any pre-specified criteria in SOP. In general, exclusion of data generated from an adequately controlled study is discouraged. On the similar note, lack of adequate control of the study parameters throughout the study can potentially render the interpretation of B data questionable. Due to time constraint, I'm not going in detail about the analytical part. However, I would like to share few observations. There is lack of adequate information or data on the stability of drug in receptor solution with highest relevant temperature. Please note that the recommendations are to maintain the membrane or skin surface temperature at 32 degrees Celsius, and to achieve that, the receptor solution temperature may be kept warmer than 32 degrees Celsius, and that temperature should be used to demonstrate the stability. Another aspect is about the matrix effect for IVPT samples in terms of preparation of calibration curve and quality control samples. Ideally, only the underneath of the skin portion should be considered. Selection of quality control samples is self-explanatory, therefore I will not go in detail. To conclude my presentation, I would say a controlled pivotal study using a well-developed, optimized and validated IVRT or IVPT method would be a promising alternative method to demonstrate bioequivalence for topical products. Proactive consideration of the observation discussed in the presentations are encouraged to improve the quality of future submission. This can lead to reduction in number of review cycles with faster approval process. I have two challenge questions. My first question is, for USP 1724, the sampling is generally performed over a blank time period to conduct IVRT study, and there are four options: A, 1 to 2 hours, B, 2 to 4 hours, C, 4 to 6 hours, and D, 6 to 10 hours. If you pick the answer C, 4 to 6 hours, you are correct. And the next question is whether this statement is true or false. The comparative permeation profiles from 1 to 2 donors are considered sufficient to demonstrate adequate IVPT sensitivity and selectivity for method validation. If you mark the sentence as false, then you are correct. With this, I would like to thank all of those who have contributed directly or indirectly for this presentation. Last but not the least, I would like to thank all of you to patiently listen to my presentation. If you have any question, please feel free to ask during Q&A session and I will be happy to answer. Thank you.